The ApiNATOMY toolkit is undergoing a radical overhaul. This is a status update on the prototyping of this toolkit. We are overhauling the toolkit to enhance formal knowledge exchange between two biomedical communities, respectively typified by the clinician and the physiologist. ApiNATOMY is now geared to directly facilitate this kind of knowledge exchange. But why is formal knowledge exchange between clinician and physiologist important? Answer: while there is considerable interdependence between clinical practice and physiology research, the vast proportion of knowledge exchanged between the two communities takes the form of free text. This type of exchange, however, is particularly inefficient if we aim to study, on a broad scale, complex physiology mechanisms that are clinically relevant to a disease, a drug, or a medical device. In particular, to ensure a more efficient knowledge exchange, we need to empower clinicians to author more explicit definitions of diseases. For example, a clinician should be provided with tools to clearly draw up the key mechanistic events in a disease. And we need to empower physiologists to classify these disease definitions based on explicit physiology principles. For example, a physiologist should be enabled to clearly distinguish a disease from another on the basis of the physiological context of the disease definitions provided by clinicians. This kind of focus forces us to pay attention to knowledge management strategies and metadata standards for resources in physiology, particularly modeling, where formalization of knowledge representations is essential, and how these strategies and standards can be leveraged to interoperate with those in the clinical space. The question then becomes, how may physiologists leverage clinical knowledge in terminologies like SNOMED, or the International Classification of Disease, or CDISC, which organize clinical data, and use this knowledge to find physiology models relevant to a particular disease? The short answer is, we cannot do this at a significant scale right now. But the physiology community can help by laying down a reference map, a scaffold of physiology knowledge against which terms from these disease terminologies are defined by clinicians. And while we're about it, organize physiology resources onto the same map as well. Let's take diabetes mellitus as an example where a physiology reference map is crucial. When prescribing, clinicians have to take into account that the disease has a major impact on drug handling by the body, directly influencing pharmacokinetics in the liver and the kidney, and by damaging the autonomic nervous system, pharmacokinetics of gastrointestinal absorption and cardiovascular distribution. But knowledge of the clinical implications of measurements, such as basement membrane thickness, intestinal transition rates, and drug permeabilities across endothelial or epithelial surfaces, is not recorded in computer-readable form. Therefore, such knowledge is neither findable nor available to inform the development of, say, a physiology model to help clinicians prescribe the right dose for a patient with diabetes. Furthermore, to build such a model, experimental or clinical data relevant to derive measurements like transition rates or permeabilities need to be found by the physiologist. Data such as concentrations of solutes across a particular membrane, as well as membrane-specific diffusion coefficients, surface areas, and thicknesses. Without an independent knowledge scaffold that relates these diverse datasets to permeability measurements, it is very difficult to automate the smart searching of physiology resources relevant to this clinical goal. And the provision of automated smart searching of clinically relevant physiology resources is key to incentivize the clinical and physiology communities to invest in interoperable knowledge management strategies and metadata standards. It is this kind of challenge that motivates the ApiNATOMY overhaul. The ApiNATOMY prototype now provides tools to build reference scaffolds of physiology knowledge. A physiology scaffold consists of three main types of entity: processes, graphically depicted as an edge; structures, depicted as a rectangular shape; and hexagons, representing measurements of state or rate associated with structures or processes. Structures play three key roles in a scaffold, namely as materials, as location, that is, sites or compartments for materials, processes, or measurements, and as conduits for processes. The notion of a conduit is central to ApiNATOMY. A structure is said to be a conduit to a process, that is, it is said to convey a process if the structure is a location to the process and is required for the process to occur. For example, an enzyme is a conduit to a molecular conversion to occur. A membrane channel is a conduit to an electrical current to flow from the inside of a cell to the outside of a cell. A tendon is a conduit to the transfer of stress energy from muscle to bone. Or a trachea is a conduit to the mass transfer of air between the outside of the body and the lungs. The data model allows for considerable eloquence in describing structures. In particular, there are 10 things a structure can do in ApiNATOMY. A structure can have a rotational symmetry and therefore an axis of rotation, consist of ordered layers of structure parts, have terminal borders orthogonal to the layers with polarity, bear distributions of scale for length and layer thickness, be annotated with terms from reference ontologies such as the foundational model of anatomy, or identifiers from reference databases such as Uniprot, fall into one of 3 topological categories, namely tube, such as the membrane channel, the proximal tubule segment of a nephron, a segment of spinal cord, bag, that is Bowman's capsule, the whole of the pulmonary epithelial tree, a gastric pit, or cyst, such as a cell at the micrometer scale, the peritoneum at the meter scale, or an ovary at the centimeter scale. Have other structures as materials or parts. Convey processes of flow of its materials. Bear measurements about its materials or its processes. Participate in assembly operations over their terminal borders, that is segmentation operations where a tube plus a tube creates a tube, a bag plus a tube creates a bag, and a bag plus a bag creates a cyst, as well as assembly over their outermost layer, to which we refer as a coalescence operation. In this diagram, we come across a coalescence example where a 3-layer nephron segment tube consisting of A, urine, B, epithelium, and C, basement membrane, fuses with the latter layer within a blood vessel segment consisting of a 3-layer tube made of E, blood, D, endothelium, and C, basement membrane, to create a 5-layer coalescence. So, let's see what we can already achieve with ApiNATOMY entities using the updated toolkit prototype. We can create canonical trees. For example, a canonical urinary tree can be assembled from a segmentation operation along an advective process of urine flow from the Bowman's capsule leaf to the right to the urethral root to the left. Each node in a canonical tree is associated with branch emission probabilities, and each conveying conduit with distributions of length and thickness. Terms from external reference ontologies are applied to semantically label both individual segments. For example, this bag is annotated with the term Bowman's capsule, as well as assembled concatenations. For example, the upstream assembly from the distal convoluted tubule upwards is an object of type bag labeled with the term nephron. We can also drape trees onto housing structures that represent organ components. For instance, we may represent the whole kidney as a coalescent assembly of kidney lobuses, with coalescence occurring over the outer cortical layer of the lobus. Here, we lay out a 2-layer bag representing the centimeter scale lobus, consisting of a cortex layer in green and the medulla layer in blue, to house regions of epithelial and endothelial trees that respectively convey urine and blood flow. We see here the assembly of conveying conduit tubes, making up the loop of Henle, the proximal tubule, and Bowman's capsule. Now, we lay down portions of the arterial vascular tree, ending with the interlobar artery, and now to the venous tree, stellate vein to interlobar vein. The leaves of the arterial and venous trees are then bridged by the glomerular capillary, efferent artery, and cortical peritubular capillary. We now prepare the ground to coalesce the peritubular capillary and the proximal tubule by aligning the corresponding conveying conduits. Assembly can also be carried out as a coalescence over the outermost layer of structures. Here we focus on the sharing of the outer basement membrane layer of the peritubular capillary, right, and proximal tubule, left, to explicitly declare that, based on the thickness scales of these tubes, blood and urine layers are within diffusion distance of each other. Satisfying this condition allows us to bridge advective processes associated with the blood and urine materials in the inner layers of the two coalescing tubes with diffusive processes for individual solutes. Looking at this complex assembly up close, we observe a two-layered cyst representing a red blood cell as a constituent material to blood, as well as sodium and its concentration measurable, all in the same blood layer. In the epithelial layer, there is a segmented assembly of two bags to create a cellular cyst, allowing for the distinction of the apical portion of the proximal tubular epithelial cell from its basolateral counterpart. This distinction allows for the assignment, for the two cellular portions, of different repertoires of membrane conduits that broker solute exchange. In this case, we observe the NH3 sodium proton antiporter on the apical membrane and the NBC sodium bicarbonate symporter on the basolateral membrane. Each membrane channel is an assembly of cytosolic, transmembrane, and extracellular segments, such that their collective outermost layer represents the protein of the channel, and the innermost layer represents the channel pore. So, what kind of resources are being integrated over the physiology scaffold? We are currently evaluating a renal knowledge scaffold to organize and integrate different types of clinical and physiology resources, including a pathway model that defines the disease process of hydronephrosis caused by kidney stone, a mathematical model of renal electrolyte handling drawn from acid-base physiology, the mining of knowledge about structures, processes, and measurables from kidney-related text in a physiology textbook, and two types of renal experimental measurement. Let's go through these examples one by one. 1. In ApiNATOMY, a disease model is a formal account of a pathological pathway. In this pathway, nodes represent measurements located in kidney structures. In this example, the pathway graph follows the cascade of events that are triggered by a change in ionic concentrations, resulting in kidney stone formation and ultimately leading to strangulation of renal blood flow due to urinary pressure buildup in the renal pelvic calcium system. Such a pathway is key to knowledge exchange between clinician and physiologist, as it provides a formal definition of a disease process that a clinician may generate in preparation for biophysical modeling by a physiologist. 2. Biophysical models leveraged to make predictions about physiology processes also take the form of a network of measurements located in biological structures. Unlike the disease pathway model we just discussed, however, mathematical equation nodes now link measurements in biophysical models. An example of a biophysical model mapped onto the knowledge scaffold studies electrolyte handling across proximal tubules and peritubular capillaries. The integration of such a model over the ApiNATOMY knowledge scaffold focuses on the linkage of state measurements to corresponding biological structures and of rate measurements to processes conveyed by conduit structures. 3. The previous two examples offered a brief look at networks of measurements that describe processes. In the next example, we rely on the renal knowledge scaffold to organize the results of text mining over 4 kidney-related chapters from the Boron textbook of physiology, kindly provided by the Elsevier Science and Technology books. Leveraging the UMLS terminology, the NIH's MetaMap tool detected mentions of structure, process, and measurement terms in the text of these chapters. The annotation of ApiNATOMY entities with reference ontology terms that are collated by UMLS simplifies considerably the mapping of kidney-related concepts identified in this textbook by this MetaMap tool. 4. The final example scenario focuses on two types of experimental measurement across a key scale divide. The first is a molecular scale experiment carried out by the Kidney Systems Biology Project, in which transcriptome expression levels were measured for each of the major nephron segments in the kidney. The second is a database of tissue-scale experiments that catalogs the location and values of measurements for structures and processes in the kidney. ApiNATOMY's integration of a wide spectrum of kidney-related information, previously dispersed over different sources and contexts, adds value to individual resources by allowing the clinical and physiology communities to pose cross-disciplinary queries that relate data and models in novel ways. For instance, physiologists may now be alerted of experimental data germane to their research effort that is detected in the scaffold neighborhood of their model variables. Clinicians may now leverage their formal definition of a disease to identify literature with an in-depth treatment of the physiology background to a pathological process. The molecular biologist may also start from a disease model, such as the hydronephrosis scenario discussed previously, to generate a list of genes whose products convey solutes implicated in kidney stone formation. And, given a biophysical model, physiologists may now determine diseases for which their model is relevant. So, what's next for ApiNATOMY prototyping? There are three main areas of priority. The first is to provide an authoring tool for clinicians to generate qualitative disease models in terms of measurements mapped to ApiNATOMY structures and processes. The second is to generate a full body graph for epithelial and endothelial processes and their conduits, covering the cardiovascular system and its coalescences with pulmonary, hepatic, and renal structures in preparation for physiology-based pharmacokinetic modeling. The third is to serve ApiNATOMY knowledge and associated resource annotation via the Fair Data Point protocol to ensure interoperability with this emerging protocol of information exchange for the biomedical community. Thank you.